Hey everyone, welcome back to biology. Today I'm broadcasting from our room 112 of Westerville South, and it's super late and it's a Monday and I'm working late and everyone else is gone. So I guess I got this entire building to myself. Today we're talking about enzymes. If you can think about Pac-Man, Pac-Man is a great way to visualize enzymes. Pac-Man eats all those little dots and does reactions, but Pac-Man is still there. So that's the first thing you want to learn about enzymes. They don't get used up. They're still there at the end of a reaction. So basics of enzymes. What are they? Enzymes are biological catalysts. Cool. Well, what's a catalyst? Great question. Catalyst is a chemical substance that increases the rate of a chemical reaction. Usually it does that by reducing something called the activation energy, which is the amount of energy that is needed to start the reaction. So it takes a reaction that would happen on its own that's either really, really slow or needs to happen at a really, really high temperature, and it lowers the energy needed to get the reaction started so it can start and keep going. So why is that a big deal? Well, most of the reactions that are happening in any of your cells or any of the cells of living things need really high temperatures to get started. Temperatures that are so high that it would damage the cell or maybe even kill the cell to get up to those temperatures. Enzymes lower the activation energy or the energy that's required to start those reactions, which means those reactions can happen at room temperature. That's the big deal. They can happen at normal temperatures that you'll see inside a body rather than super high temperatures. Here's a graph that kind of shows that process. You can see over here on the left, we have reactants. Those are the things that you're starting with. Products are what you have at the end of the chemical reaction. And the graph shows the amount of energy. So higher up on the y-axis is more energy, lower is less energy. You can see this energy from start to finish, the graph goes down. So it's going to release energy. This might be showing a reaction that we're breaking down food in our body to get energy and use that for other things. But you'll notice the graph doesn't go straight down. It doesn't go straight from the reactants to the products. It has this little hill. That hill is the activation energy. That's the energy needed to get the reaction started. It's kind of like lighting a match. The match will burn, but if you just look at it, if it just sits there, it's not going to burn by itself. You need to put a little energy into it to get that reaction started. Then once it's burning, the combustion reaction will continue and the match will continue to burn as long as it has oxygen. The extra energy you put into that match is by striking it, which actually creates friction, which generates heat, which is the activation energy to get that reaction started. Similar idea here. It's sort of like once you put in a certain amount of energy, then the reaction continues and a bunch of energy is released. What enzymes do is they lower that activation energy, which means you need less energy or less temperature to get them started, which is a really big deal because living things are typically pretty sensitive with the temperatures that they can endure. And without enzymes, cells wouldn't be able to carry out the reactions they need at the temperatures that they can survive. Okay, so that means temperature is a big factor. So enzymes can only function, let me go back. Enzymes allow reactions to happen at room temperatures, but by lowering the activation energy, that also means that they can make reactions happen a lot more quickly. So that's why they're referred to as catalysts, because they speed up chemical reactions. They make them go much more quickly. They're also not used up in those reactions. So by definition, a catalyst is not part of the reactants or the products. It just helps bring them together to make the reaction happen quicker, but they're not used up. Pretty much every reaction happening in a living thing uses an enzyme, which is really, really important because there's tons of chemistry happening in even a single cell or a single-celled organism. Almost all of those need enzymes to make them happen. Okay, how do enzymes actually function? It's all about the shape. We'll come back to this idea at the end of the notes when we talk about how you can break or denature an enzyme. But enzymes function because they are protein molecules that have a very specific shape, and that shape means that they are suited to one reaction. Usually, they're only going to be suited to one very specific reaction. That term is called specific. So enzymes are really specific. They only work on one thing. Here are some examples of enzymes. There are thousands and thousands and thousands of different enzymes. Here are just a couple. First is lactase. You may have heard of that enzyme. That breaks down the sugar lactose into glucose and galactose. Another enzyme is catalase, which breaks down hydrogen peroxide into water and oxygen. That's another important one because hydrogen peroxide can be harmful if you build up too much of it in your cells. What those two enzymes have in common is the suffix ase, ACE. Anytime you see something ending with ACE in a biological context, it's an enzyme. So if you're watching some medical TV show and they talk about something that ends with ACE, you can say, oh, they must be talking about an enzyme. And you look like the smartest person in the room. Hey, maybe you even are the smartest person in the room. Good for you. All right, here's a video. I'm not going to show this one right now. I linked it in the folder. It's under video 1. This will explain activation energy using this idea of a snowball rolling down a hill. Strongly recommend you go check that video out. If you're confused at all or you want to refresh what I mean by activation energy, especially those crazy graphs we were looking at a minute ago. Okay, how do enzymes lower activation energy? Basically, enzymes aren't used up in a reaction, but they're going to physically help that reaction happen. Sometimes that means they'll sort of grab onto other molecules and bring them together. Sometimes they'll grab onto a molecule and twist it and put tension on it and make it easier for it to break apart. But whatever they're doing, they're physically making the reaction more quick or faster when it happens. So here's a picture of an enzyme in action. This molecule over here is called the substrate. That's what we're starting on, and that's what the reaction is beginning with. You can see down here is the enzyme. So the enzyme helps the reaction happen, but we still have one enzyme when we start, one enzyme when we end. It's not used up. It's not changed. It's not altered by the reaction at all. The active site is the part of the enzyme that actually binds to the substrate in the reaction. And then the product is what we are left at the end of the reaction. So the reaction is substrate getting converted into product. The enzyme helps that happen by binding to the substrate at the active site. And you can see it's all about shape. This molecule looks like it has a really good shape for grabbing onto that substrate. Maybe it's twisting or putting pressure on it somehow that makes it easier to break it apart into the products. Oh, products again. Great. Here's where it gets a little more tricky because there are different enzymes for all different kinds of reactions. Sometimes you might have one substrate that gets broken apart into two products. Sometimes you might have two substrates that get put together to make one product. Sometimes you might even have one substrate that just gets altered to be one product that's different than you started with. The point is there are lots of different options. You can see here on the left, there's one substrate, binds to the enzyme at the active site, broken into two products. So that's a breaking down chemical reaction. On the right, we have two substrates that bind at the active site that are joined together to make one product. And it can get even more complicated than this, but just know that sometimes there are one or more substrates, sometimes there are one or more products. Here's another video. This is like a cartoon description of enzymes. Trust me, folks, this one is definitely worth your time. If you watch this video, this is video 2 in the folder. You will not be disappointed. It's one of my favorite videos of the entire year. I don't want to say too much. You will definitely enjoy it. Okay, a little bit more about enzymes here. There are a couple of different explanations for why enzymes work the way they do. This one is a little bit of an older idea. It's called the lock and key theory. And it says that the active site basically has the perfect shape to bind to the substrate, and the two come together like a lock and a key, and that's how they actually work. However, we think there might be a little bit more complicated things going on here. So a newer idea is the induced fit theory that says the active site is, pretty good, but not perfect. And as the substrate binds to it, it changes and conforms and sort of becomes perfectly shaped, but it is naturally close, just not perfect. So either way, the long story short is it's about the shape. Enzymes function because of their shape. So how do you stop an enzyme from working, or how do you break it? First of all, that process is called denaturation, or to denature an enzyme. You can say an enzyme becomes denatured due to the conditions that it is exposed to. One thing that can denature an enzyme is temperature. So, as I said earlier, all enzymes are protein molecules, and those protein molecules may lose their shape at a really high or a really low temperature. If they lose their shape, they can't function as well. If they lose their shape entirely, they won't be able to help the reaction at all, and it'll completely stop. This sort of the opposite of denaturization is this thing called an activator. Activators are just other chemicals that might bind to an enzyme that actually make them work faster. And that's also based on shape. So they'll change the shape of the molecule and make it maybe a better fit for the substrate and help the reaction go quicker. pH is another thing that can denature an enzyme. So you might have learned about pH last week. Maybe you'll learn about it next week, depending on when you're watching this video. But pH is basically a number that measures how acidic an environment is. And that just depends on the enzyme. Some enzymes function the best in a really acidic environment. Some will function best in a basic environment. Some will function best in a neutral environment. But if you take any enzyme out of its optimal pH range, it won't work as well, and it might even get denatured completely. Lastly is this thing called an inhibitor. Inhibitors are the opposite of activators. They bind to an enzyme and will either permanently bind at the active site and shut that enzyme down, prevent it from working, or they might bind somewhere else to the enzyme that causes it to change shape and morphs the active site out of the shape that it's supposed to be in. But inhibitors, one way or another, change the shape of an enzyme, slow it down, or denature it completely. The big takeaway here is enzymes function because of their shape, and anything that disrupts the shape, whether that's temperature, pH, an inhibitor, or an activator, will affect the performance of the enzyme and the speed of the reaction. And if you denature an enzyme completely, it loses its shape completely. It will no longer be able to function, and the reaction may stop altogether. Oh, that's a call. Getting a call right now from my wife. So I guess that means this video is just about over. These pictures show you what happens when you denature an enzyme. So enzymes are proteins. You can see here, this is the normal shape of the protein. If you heat it up, it'll lose its shape and becomes unfolded. And even if you cool it back down at that point, a lot of proteins won't go back to the state they're supposed to be in. Same thing down here. You can see here's an enzyme. The active site is well fitted for the substrate. If you heat it up, it loses its shape and can no longer bind. Here's another visual of what it might look like when an enzyme gets denatured. It changes shape. The active site changes shape, maybe because of heat or pH, and it can no longer function because it doesn't have the right shape. Lastly, in the folder, you'll see an Amoeba Sisters video. Oh, she's texting now. I really better wrap things up. You can see an Amoeba Sisters video about enzymes. It's an excellent review of the things we talked about today. And if you're confused at all, I direct you to watch that video. Thanks for watching. And if you have any questions, please let me know. And don't forget to keep on swimming.